Welcome to Study Life. We are seeing our chapter Enhancement in Food Production and our topic is Applications of Tissue Culture. When we see tissue culture, we have four applications. The first application is micropropagation. The second one is production of disease-free plants. Third one is production of secondary metabolites. And the fourth one is somatic hybridization. So in this video, we are going to see the first two applications. First one is micropropagation. As the name suggests, micropropagation. It is an application of plant tissue culture where large number of genetically identical plants can be produced. When I say large number of genetically identical plants, means they will be all obtained from the same plant. So they are large number of genetically identical plants. Now for this, what we do is, this is our tube in which we have taken nutrient medium. This is our nutrient medium. As discussed earlier, this nutrient medium is going to be slanting so as to increase the surface area for absorption, and here we would be taking explant. This explant is shoot apical meristem. This explant is shoot apical meristem is used as an explant, and we have to maintain aseptic conditions. Aseptic conditions means sterile conditions. So when we say we have to maintain aseptic conditions, they'll be covered with a cotton. So for this micropropagation, we take shoot apical meristem as an explant. By adjusting all the growth hormones and nutrient medium properly, what do we get? After some time, we see that what happens is, in this same tube, we will find many shoot apices. See, this was our shoot apical meristem. So we will see there is formation of many shoot apices or apical buds. Now these small, if you can see, these are called as shoot apices, or they are called as apical buds. Shoot apices or apical buds are obtained. So this is after using the nutrients, the cells of this shoot apical meristem starts multiplying, and we have many shoot apices or apical buds which are growing. Now these shoot apices or apical buds, let us remove them out and see how they look. These small are shoot apices or apical buds. They can be used as micropropagules to obtain large number of genetically identical plants. So they are going to form micropropagules to obtain large number of genetically identical plants. All the plants will be genetically identical. So let us see the advantages. Why this micropropagation is useful. Now this micropropagation, the first advantage is we get large number of genetically identical plants. The second one is those plants which were season dependent. Now because we are growing them in the tube, so season dependent plants, they become season independent. Season dependent plants become season independent, can be grown. And here in this, we can grow, when we say we have genetically identical plants, which means we can get clones of the plants. So second point would be we are getting clones of different plants. So third point is rare plants can also be produced. Now these rare plants also include economical plants which give us a lot of benefit. They can be produced by this micropropagation. Now let us see an example. In this shoot apices or apical buds, if we say we had taken one shoot apical meristem and from that we have got 100 shoot apices or 1000 shoot apices, we can obtain so many new plants. So we'll say this micropropagation requires small space. So our storage becomes easier. This storage, can you imagine a test tube containing 1000 plants? So micropropagation, it requires less space. So storage becomes easy. Now this was our first application. Now let us see the examples of this. Examples include banana, potato and orchids. They are widely grown by using this technique of micropropagation. We have examples like banana, orchids and potato. They are grown by the technique of micropropagation. So this was our first application of plant tissue culture. Now our second application for plant tissue culture is production of disease-free plants. Now here we are going to make disease-free plants. Now for production of these disease-free plants, I'll give you an example. Suppose we have a diseased plant, a plant which is having a certain disease. Now from that diseased plant, it is possible for us to recover a healthy plant. How can we obtain? We can obtain it by this technique. For production of a healthy plant from a diseased plant, healthy plants can be recovered from a diseased plant by using shoot apical meristem. We are using shoot apical meristem as an explant. Now, if you know what is shoot apical meristem means it is the shoot which is called as the stem, the apical part which means the top part, and when we say the word meristem, meristem means meristematic cells. So we have a plant whose top part and those cells are made up of meristematic cells which are newly formed cells. Now, why do we take this shoot apical meristem? The only reason is this shoot apical meristem is newly formed. The cells are newly formed, and because they are newly formed, they are free from infection. So though the entire plant is infected, this shoot apical meristem, because it is newly formed, it is not going to have that infection and it will be disease free. So in that part, we can cut that shoot apical meristem, we can grow and we can recover a healthy plant. The examples for this is banana, potato and sugarcane. Banana, potato and sugarcane. These are the examples for production of disease-free plants. So in this session, we have seen two applications of plant tissue culture. For more videos, please stay tuned to Study Live. So students, you can definitely try the upcoming questions to see how these topics are well understood. Do try them. That's all. Thank you.